In Array Studio, views are ways to visualize the underlying data loaded into Array Studio memory. Views are associated with objects, so filtering the object will change the data being displayed in the view. The table view shows the measurement data for a given object, but you can't change the object value. You can change annotation or design metadata except for the ID column. To add a new view, just right-click your object. Depending on the object type, up to 40 different views are available. Let's create a boxplot view, which shows the range of delta CT, or relative abundance, of each gene for each sample. We can filter the data based on metadata, such as the control or treatment group. This will filter the boxplot view and also the table view. Views are highly customizable. We can rotate the labels to make sure we can read every sample's label. We can also assign colors to each box plot by tissue group. And we can trellis the data to separate the data into two charts based on cell type. Now let's add a dot plot view. For each gene, the range of values for each sample is being shown. Let's color by tissue and group. We can change the shapes, increase transparency, and add a little jitter so that our samples don't overlap entirely. We can change the annotation ID column to gene name. We can see that some genes are clearly differentially expressed between breast and lymphoid tissues. Now let's add a variable view. Again, let's change our titles from Ensemble ID to Gene IDs. In the variable view, a separate chart is created for each gene. We can group our samples by design metadata. You can filter by gene name, for example, to quickly find a gene of interest. We can see that treatment reduces expression in breast relative to control, but treatment actually increases expression in lymphoid. Within the variable view, we can also change the details of how the data are plotted. For example, we can show the data as a violin plot. Now let's filter for a set of genes. Hold down Control to select multiple genes. We can view the expression relationships between these genes with the 3D variable view. Here, to view the gene IDs, we will need to change our ID column. Here we can see how each of these 3 genes changes its expression between our sample groups. We encourage you to explore all the different views and see how you can customize them to most efficiently explore your data.